welcome to the special session of our mini colloquium out of equilibrium physics in biology. This is the special sessions and in which we will have 13 flash talks, but more information about that work is already uploaded in the webpage and corresponds to 3 pre-recorded talks and 10 posters. And after these 2-minute sound bites, you will have time to discuss individually with the speakers of each of them in a breakout room. All of you by now are made co-host, so you will be able to move from room to room freely, depending on your interests. But if you find any trouble, please contact Juan through the chat. That is going to take care of that. So we can start now, and we have Cristobal Diaz. So the screen is yours. Go ahead.
Okay, thank you, Laura. So thanks for letting me present my presentation, So what I will talk, it's a little bit about what I did. I did this numerical simulations for the cell-mediated self-assembly of scaffolds. So scaffolds are usually the base of when you want to build artificial organs. But as you can expect, and you can see from this picture in the left, they are usually done outside the body, as a 3D printing or as a donor using a dead organ. So what our experimental collaborators from Antwerp did is to, they did an experiment where you have colloidal particles and you covered these colloidal particles with some antibodies that can choose the own cells of the organ that will grow to self-assemble and use these cells to self-assemble. And as you can expect, you have a lot of possibilities. You can try different ratios and fractions and size of particles. So what we did, and you can change the slide now. So we did a numerical simulations. So we created a model, as you can see here. And we have the main, I'll show you the main mechanisms. We have these adhesive domains in the colloid, and cells can adhere to these domains. You also have adhesion between cells, and then you have the main mechanism where you have a cell-mediated bound. And so what we proposed, we validated this model, and what we propose is that we can, what happens if we block the cell-cell adhesion mechanism? And as we see, and you can see in the plot, the blue ones are the ones with the blocked mechanism, and you can see that we improve the cell-mediated aggregation, improve the size of the scaffold. And so we send this to the experiments to test, and they also show the same behaviors. We can kind of a validation of our model. So thank you. I hope I didn't take too much time. I think you're mute, Laura.
Yes, Laura, you're mute. Laura.
Sorry, that you did perfectly on timing. Sorry, I don't know what my computer is doing. So let's go to our next speaker, Danilo Silva. And yes, I'm going to share my screen and you can go on.
Hello everyone, thank you for having me. My name is Danilo Silva and I'm a PhD student from the University of Lisbon. So my work here is about the discontinuous shear thinning behavior of soft particles in 3D microchannel. Here we were motivated by the fact that in many applications like microfluidics, for example, we have systems composed of a large number of particles. And more often than not, these particles are flexible. And this flexibility can in turn affect, for example, the hydrodynamics of the flow in this type of systems. Another application, for example, if we look at cells, we can see that cell deformation is a very important quality in many biomedical devices. So in light of this, what we did was we used a multi-component, multi-phase model using the lattice Boltzmann method, where we simulated one particle, the flexible part gas being one fluid, and the surrounding medium being the other fluid. And we were able to expand this to a 3D model using higher order lattices, and giving a choice of parameters, we are able to frustrate the mechanism of coalescence between the droplets. So then what we did was,
sorry.
Okay. So then what we did was we simulated a large number of particles in a cylindrical channel driven by a force with neutral wetting. And to vary the flexibility, we used the surface tension as a tunable parameter. So then we looked at the relative viscosity as a function of the capillary number for various surface tensions. And we can see that they collapse. We can see a discontinuous shifting behavior on the curve, and we can see that the curves collapse into one master curves. We suggest that the capillary number is the primary variable that we should be looking at when determining the flow response to deformation. I think that's it. If you want more questions, just talk to me later.
Thank you very much. That was perfect. I'm very good on time. So we will have now our last pre-recorded talk. I'm going to share this. So Berta Tinao is now presenting. Berta, whenever you are ready. Berta?
Yeah, sorry, can you hear me now? Sorry, okay. Is everything working?
Everything is working.
We can
Okay.
hear you, yeah.
Yep.
Okay, so hello all, my name is Berta Tinao and I'm from the Autonomous University of Madrid. I'm doing my PhD there as well. So as you could have seen in my video, we try to shed light on how cells are able to move through complex environments such as our veins. So we use a synthetic model system composed of a stratigrating coated ferromagnetic particles on top of biopolymer hydrogels. So we actuate these particles with an external rotating magnetic field, and through friction against the substrate, these particles are able to translate. So to have an idea of how much friction the system has, we define a dimensionless parameter, which is called a rolling parameter, for which the 0 value means no friction at all, and 1 value means a perfect rolling regime. So if you could change, thank you. So briefly talking about the results that we got, we've observed that the sortaglin strongly interacts with the polyacrylamide and collagen fibers. So we had to coat the particles with a design lipid coating. In the case of polyacrylamide gels, electrohydrodynamics theory suggests the appearance of a lift force that reduces the friction between the particle and the substrate. And in the case of collagen, we also had to consider the interaction between the lipid, the head of the lipids and the collagen fiber. So we added the pegylated lipids. And as we could see from the results, we obtained that varying this amount of pegylated lipids, we can modulate this interaction with the collagen fibers as well. So thank you very much. Sorry for the throwbacks, and I'll be glad to answer any question.
Laura, you are...
Yeah, it's fine. We move now
left
to
it
the...
open, so.
Eh?
Left it open.
Yep, okay. So we move now to the posters. These were the pre-recorded talks. So the posters are easier because it's a single PDF, so I can share with you only one more time, and this should be fine. Can you see well?
Yeah.
Yeah. So Amit's vision is presenting now.
I can't see actually. I can't see the screen.
Oh, you can't?
It's not loading for some reason.
Oh. You cannot see it?
No.
Okay, I'll do it again. Stop share. Share screen. You can see it or not?
Right now I can see. Yeah.
We can see it, yeah.
Yep.
But not the
But
full,
not the full screen.
yeah.
But not in full screen.
No? Okay, I'll do that. I'm gonna stop sharing, enter the full screen mode and do it again, right?
Yes.
Okay. Sorry for this.
We are learning how to handle these things online. So next time, each one will share the screen and will be much, much faster.
Next time, I hope we can do physically.
Yeah.
Yeah, hopefully.
Hopefully. Let's see if now this works.
Yeah, this works. Yeah, thanks.
Perfect. Thank you. Sorry. Go
No,
ahead.
it's perfect. Thank you. Hi, hi everyone. My name is Amit. I'm a postdoc at Institute Curie, and thank you all for giving me this opportunity to present. So many biological functions like adaptation, proofreading, motion of motor proteins require a finite rate of energy input. Hence, the dynamics describing these systems has to be necessarily out of equilibrium. In contrast, there are also processes which are non-equilibrium at fast time scales, but can be effectively described at a slower time scale as an equilibrium through an equilibrium model. So what I'm going to talk of, what I present in this poster are through a toy example, I studied these two different aspects. And can you go to the next slide, please? So what we study is we consider two particles driven by two different temperature baths interacting with each other. And to analyze the effective equilibrium limit, we introduce a timescale of the timescale separation between them through a variable tau, which essentially makes the variable y much faster than variable x. And to compare with various models like that of active particles and that of switching fluctuating potentials, we try two different values of epsilon is equal to half and epsilon is equal to 1, which lead to different results and is one of the main points. So the function we are interested in studying is the variance of the particle x. The next slide, please. For feed-forward dynamics, what we show is that if we calculate the variance and the entropy production rate for the feed-forward dynamics, we find that at a given time scale, the variance increases with the increase in entropy production rate. For the feedback case, no such one-to-one mapping exists between the entropy production rate and variance. However, a one-to-one mapping can be obtained by calculating the minimum variance that can be attained for a given entropy production rate and vice versa. We find that this minimum variance is a monotonically decreasing function of the entropy production rate. We then go on and calculate the hidden entropy production rate in the limit, in the effective equilibrium limit, which is obtained by taking tau going to 0. And we find that the similar relations between the hidden entropy production rate and the variance exist in the effective equilibrium limit. Thank you.
Thank you. Okay, our next speaker, Jenilson Carballo.
Yes, I'm here.
Thank you. Whenever you are ready.
Okay, can I start
Yeah,
now?
sure. Go ahead. Yeah.
Okay, thank you. Okay, good morning, everyone. My name is Jenilson Carvalho. I'm a PhD student from the University of Lisbon, Portugal. My presentation is about a work that started in collaboration with the experimental group of the molecular biology at Nova University of Lisbon. In this work, we studied the deformation, the epithelial tissues of Drosophila larva after a laser cut on the cell membrane. We want to characterize the mechanical response of the tissue in relation to its most relevant physical parameters, such as viscosity between the cells and the friction between the tissue and the substrates, for example. For this, we developed an analytical and numerical theoretical model for a continuous and viscoelastic tissues in compressive and thin. Initially, this very thin cell tissue is only stretched under sigma radial tension. Then a circular cut is made in the central region of the tissue. We solve the equation of the numerical and the what for the tissue to relax. We analyze the evolution of the tissue displacement and velocity for different times. We also analyze the normalization of the displacement and velocity for different parts of the tissue. We investigate the competition between the viscosity due to interaction between cells and the friction between the cells and the substrate. Next slide, please. Our results show that there are two well-defined regimes dependent on the lambda value. For lambda values, low lambda values, the friction is relevant on the system. For larger lambda value, there is a domination of a viscous interaction. Thank you for attention.
Thank you very much. We move to our next speaker, Jose Martin Roca.
Yeah.
Whenever you are ready.
Okay. Hello, everyone. I am Jose Martin, PhD student of Chantal Valeriani in Complutense University of Madrid. And I'm going to talk about characterization of motility-induced phase separation, so-called MIPS, in a system of active Brownian particles. To study this, can you go to the next slide, please? Thank you. To study this kind of phase separations, we compute a phase diagram, like we can see here, density versus Peclet number, that show us the value of these two parameters for which we can find MIPS or not. The empty dots are homogeneous phase, and black dots are mixed phase. Okay, one of the main point of our work is to study the effect of varying Peclet changing different parameters, because like you can see at the top in the left, yeah, here, thank you. By definition, Peclet number can be varying fixing all parameters by one, and then you can compute the phase diagram, but you have a lot of combinations, and we study how the phase diagram change depending of which parameter you want to vary, to change. That's one of the main points, and the second one is to study the effect of change for the same way to change the Peclet. Use another interparticle potential. This only make the particles, the colloids, harder or softer. And the last point is another way to compute this phase diagram, that usually we use the local density, like we can see using Brownian tessellations, but we propose to use negation parameter because it's easier to compute that Brownian tessellation. Well, that's more or less everything. It's a
Thank you.
Work. Thank you very much.
We'll move to our next speaker, Julian Coetzee, whenever you are ready.
Hi, you can hear me, right?
Yes, and we can see you.
Ah, great. So hi, my name is Julian, and my poster is about quantifying relative probabilities of stochastic trajectories. For this, we consider tubes around smooth paths. So if you look to the plot on the right, you see a black line, which is a reference path, a smooth reference path. And the tube around it is shown as a gray shaded area. And you see the x-axis is time and the y-axis is position. And of all the stochastic trajectories that start inside the tube on the left side, some stay inside the tube at all times, like the blue one, but there are a lot of them, of course, leave at some point, like the orange one. And the probability to remain inside this tube can be characterized, of course, by the exit rate with which stochastic trajectories leave for the first time. This indicated by the green arrows. And the interesting thing here is, if you look to the equation on the left-hand side, the exit rate for asymptotically small tubes, so if you shrink the gray area down to radius 0, then the difference in exit rates is related to the stochastic action Lagrangian, which is known to characterize ratios of path probabilities. And we think this is a remarkable equation because it relates the action Lagrangian on the left-hand side of the equation to an observable exit rate, which is a very intuitive way of characterizing path probabilities. And using this relation between the exit rate and the stochastic action, we have measured relative path probabilities experimentally without fitting a model to the data. And also we asked what happens if you don't take the limit. And so we removed the limit r to 0 on the right-hand side, and we calculated the next order corrections to the stochastic action Lagrangians. So the big picture is that we consider tubes as surrogates of individual trajectories. If you want to know more, then have a look at my poster or look at the references. Thank you.
Thank you very much, Julian. We move to our next speaker, Juan Pablo Miranda, whenever you are ready.
Okay, do you hear me or do you see me?
Yes, we can see you and hear you clearly.
Okay, thank you very much. My name is Juan Pablo Miranda. I'm working with Chantal Valerian and Valentino Blanco in the Complutense University. This is about my master thesis, and we are studying the structural features of an active ring polymer and then confinement. Next slide, please. We propose a system consisted by a polymer, it's a Brownian polymer who has its activity implemented in the parallel to the skeleton of the polymer, like the next neighbors, and this polymer is confined between two parallel plates. This is a theoretical system, so we have studied the Langevin dynamics of computing simulation using lamps. And next slide, please. And the thing we want to characterize first was the structural behavior and the configuration and conformation of the polymer to this to achieve this. We have studied the radius of gyration of the polymer at distinct activities or platelets and in different distance of the confinement plates. And the main results we have seen is that the activity induces expanded states of the polymer, like the polymer collapses at low activities, but at higher activities it expands, and also the confinement induces expanded states, and this is a competing regimes we have seen that we have new results. So I think that's it.
Thank you very much. We'll talk to you later. Martin Bravo is presenting next. Manuel, whenever you are ready.
Hello, can you hear me and see the
Yes,
slide?
we can see you and hear you.
Thank you. So let me introduce myself briefly. I'm a PhD in Tenerife, the Canary Islands, Spain. We are working using a coarse-grained model based on rigid bodies for the study of icosahedral capsize of viruses. Here we have a rigid body with proper axis and a distance between. So we have developed a coarse-grained model which is very interesting by one reason, and it is that it provides us a full Hessian matrix for the binary interaction. That's remarkable, since so far we haven't found any other model which provides such a thing, which is very useful for the analysis of the normal mode. We have applied the normal mode analysis for the empty capside of the satellite tobacco necrosis virus, based on the work of Teichmann and his thesis. Despite the Teichmann thesis and his paper was an atomistic calculation, we have been able to fairly reproduce the normal with a fit about 5% of error. But here is the surprise. Do you remember what we are talking about the full geometrics? Well, so we have found that for pentons, that is the dodecahedron, we have a latent instability. We don't have a minimum for the binary interaction. But which agrees with the experimental observations, since the satellite tobacco necrosis virus can be only self-assembled with trimers, which only takes place in presence of genetic material. So despite we are using a coarse-grained model and we are performing just a normal to error, the stability of a full capside. If you have any questions and want to know more about our work, you have here the DOI and a link to our paper. If you have curiosity about the movements, there are videos available as supplementary material.
Thank you.
In active matter, people are always interested in understanding the structural and the dynamical properties of the collective systems. But we are particularly interested in the soft interactions because of its relevance in the biology and especially in the collective behaviors of cells in tissues. So for the softness, we have introduced here the parameter alpha, where alpha, which you can see in the potential. So which is written on the right hand side. So which is, as you can see in the plot, for if alpha is equal to 6, the overlap between the particle is very less relative to when alpha is equal to 1. It means as we are going from alpha equal to 6 to alpha equal to 1, we are making our particle more and more soft. So doing this transition, we want to see how the structural and dynamical properties of the system will change. Can you move to the next slide, please? Laura, yeah, thanks. So as you can see in the left hand side, the structural properties in the first is a XY coordinate particle snapshots, where color map is the quantity which is the local orientational order parameter has calculated. It shows that while changing the softness of the particle, the structure of the phase separated systems has changing. So we expect that the dynamic within this phase separated system will change. But as we did a study, so you can see in the dynamical properties in the inset, which is with the red dots, that effective diffusion of the particles is approximately the same. So we wanted to make sure that this behavior is arising because of the activity-driven phase separation, or it's just the behavior which is arising because the system is dense. So we did a simulation where system has no phase activity into it, but we found that that was a function of a softness, which let us conclude that this the phase separation in the system, the structural properties do depend on this parameter that we have introduced, which is called the softness, but the dynamical properties of the system does not depend on that. It somehow seems to be the property that is originated from the fact that the system is actively phase separated. So for more information, you can look at our poster. Thanks.
Thank you very much, Monica. We'll find you. We move to our, it's not the last, because later our last speaker will share his screen by his own. So, but now Patricia Esteban Infantes will be presenting.
First, do you listen to me?
Yes, perfect.
I want to apologize if my connection is a bit slow. So having said so, I start. I'm Patricia from Complutense University of Madrid. And for my master thesis, I've studied dynamics of active filaments under confinement. Can you change to the next slide, please? So we have simulated polymers with different number of monomers in corrugated channels, as shown in the left picture. We vary the minimum and maximum radius of the channel, which creates an entropic barrier as defined in the equation below the figure. And we take the active force of the polymer tangent to the backbone of the the polymer. So we tried to characterize the dynamics by means of the mean square displacement. And when we analyze the graphics in logarithmic scales, we found 3 different regimes. And we used the last one to compute the diffusion coefficient. But we also have some interest in the crossover times, the green triangles, that mark the transition between the 3 dynamic regimes. So can you change the slide? What we found about the diffusion coefficient is that it followed up our law with the active force, and that it remained constant with the number of monomers, at least for the Peclet values that we were able to explore. We also found that the diffusion coefficient decreased with the entropic barrier above a certain threshold. And this is because the entropic barrier makes it difficult for the polymer to pass through the bottlenecks of the channel. And this effect is less visible the larger the Peclet values. So the threshold of entropic barrier is larger for larger values of Peclet. As for the crossover times, we found that they were all independent of the entropic barrier, and they follow decreasing power laws with the active force, because the active force accelerates any dynamic change in the polymer. But we also find that the long crossover time, the second one, also depends on the number of monomers of the polymer, because in the end, it is the time the polymer takes to fully expand in the channel and start to explore through the bottlenecks. So thanks for your attention.
Thank you very much. So we can have now, I will stop sharing so that Diego, you can share yourself.
Yeah, thank you. Can you hear me well?
Yep.
Okay, nice. So this is my screen. Can you see it?
Yes.
Okay. So thank you for having me, and thanks to the organizer for this colloquium. So the work I'm presenting here is a part of my PhD with all these people you can see here. Sorry for that. Well, we are here in the Universidad Autónoma de Madrid, and in this work we have studied proton transfer reaction in DNA in guanine cytosine base pairs. Basically, this is a problem which has been studied in detail in vacuum. That's in gas phase, right? And this here is the summary of the result. Basically, you see that we have 2 protons that can jump from one side to the other, right? This here is just the distance, the position of this proton here. And what you see is that this proton can actually, it has 2 stable positions, configurations. And this configuration here is a rare tautomer, which it yields substitution mutations in DNA, which is the interest of this problem. But this problem hasn't been studied with enough detail, accounting for the full complexity of the biomolecular environment, which is what we have done here with a recently developed QMMM method with Amber and Fireball, our local orbital DFT code. So this is just a snapshot to give you an idea of the complexity that we are taking into account, right? Basically, we have this result in vacuum, and we have comparisons with more accurate methods. But when you take into account the environment, you can see that the metastable configuration disappears. So in some sense, the conclusion of this QMM study is that the environment makes the ray tautomer unstable, and thus it protects against spontaneous mutations, the genetic code. No? We also computed some free energy maps taking into account the positions of the 2 reactive protons, and we studied further some aspect of the reaction mechanism, so that if you have any questions or comment later, we can talk about it. So, but that's all for now, so thank you very much for your attention.
Thank you very much. I want to thank all the speakers because you did it super well and fit the time very, very well. So we can now, if you can turn on your cameras, we can now take a picture of all of us together and later go to the breakout room. Thank you. Juan, can you take the picture for us? Thank you. Juan, stop the recording.